Welcome, fellow beekeepers. This is Jamie Walters, your live webinar training host. I'm pleased to share this educational webinar in hopes to make you a better beekeeper. Live sessions are now recorded on the fourth Sunday of each month at 7 o'clock p.m. through Zoom conference call. You're welcome to reserve a ticket at www.eventbrite.com. Search for Ohio State Beekeepers Association's events and click Tickets. Register by filling in your name and email address, and a confirmation email will be sent to you with a Zoom link. Each training session is recorded and posted on OSBA's YouTube channel. You're welcome to subscribe and be sure to click the bell to be alerted each time we upload a new video. Each month's speaker and presentation will be posted on OSBA's Facebook page and on our website at ohiostatebeekeepers.org. You're welcome to subscribe to the live webinar training news group to be alerted at the beginning of each month. If you have any questions, please feel free to contact us at live-webinar-training@ohiostatebeekeepers.org. And now on to our previous recorded webinar. And tonight I am very grateful to have Dr. Hongmei with us, and she's going to talk about the lethal viral infections. So Ms. Hongmei?
Thank you. My name is Hongmei Li-Bylarlay. I'm an assistant professor of entomology at the Central State University. So welcome all of you on a drizzling, cloudy Sunday, but spring is coming. I am very excited for this field season. So we just got our nukes the other day, so we're very excited to get more bee colonies going for our research. So today's topic, I'm going to focus on viruses. We'll talk about a lot of information in honeybee viruses. In second part, I will talk about a recent paper we just published about IAPV virus. So first part of work, we're going to ask the question, why do we need to learn about viral transmission in the honeybees? What information will we know? Why are they important for beekeeping? So honeybee viruses, or any viruses, like right now we're in the middle of COVID pandemic, are caused by coronaviruses. So, and bees as well, they have bee viruses everywhere. So these viruses are obligate intracellular parasites. Basically, they can multiply and replicate themselves in the living host cells. So like coronavirus will replicate in human host, honeybee viruses will replicate in the honeybee host. So they use, for example, the honeybee virus, they use the host cells and manipulate the metabolic machinery of the cell, and they would produce more variants in the host body. In order to survive, a lot of viruses, they must have ways to invade hosts, and they will be transmitted from one host to another. This transmission process determines the persistence and the spread of viruses in a population. So for the first part of viral transmission, we're going to talk about 4 parts. The first is vertical transmission in the honeybee colonies, and then horizontal transmission, and then other vectors and factors, and the last part is beekeeping management. How can we do to make it better, stop the transmission? This is a picture to show male drone on the left. So that's a drone honeybee, and a queen on the right. So there is a venereal transmission when the queen bee was in the mating flight in the air of the DCA area, the drones were trying to find the queen and mate with her. During this time, there's a possibility to transmit viruses from the semen spermatheca to the queen. So that's one type of vertical one type of transmission. And after they mate, the queen bee, as everybody know, will lay eggs. The queen bee will go back, will fly back to the hive she was in and start laying eggs. So the vertical transmission means the viruses can be passed from the queen to the eggs, and then from the eggs to the larva, to the pupa, and then to the newly emerged adult workers. So by this route, that's a vertical transmission route. So viruses can infect ovarian tissues of the queen and then disseminate in developing eggs before oviposition. The vertical transmission can be presented in a lot of tissues. So we're looking at these tissues listed, the queen's feces or hemolymph, the gut, ovaries of the queen, spermatheca, the head or eviscerated body. And then I'll show you the percentage of virus positive samples, N equals 10 for each tissue. Now, this is a paper has been published in 2006 by Chen et al. So this was the research carried out by Judy Chen at the USDA Maryland lab. So when we look at different viruses here, ABPV, that's acute, that's the acute bee paralysis virus. We see no presence in any of this tissue. But the black queen cell virus, we see 100 in the feces, 100 in the gut, 70% of ovaries, etc. So you can see from this data, the deformable virus is pretty much everywhere. Some of the CBPV and some of the SBV. So that's a chronic bee paralysis virus. The KBV is one of the other virus here. The SBV is the try to remember. So it's been a while. So okay, I'll get back to the SBV and KBV full names. So the paper also tested different tissues by the gel pictures. They did run gel pictures to make sure the viral infection was presented in all the different tissues here. So the bottom part are the control gene marker. On the top is deformable virus. They had designed to purposely target a certain area of the deformable virus, a fragment, and they can run PCR. This is polymerase chain reaction, and all these binds, you can see a line, like really shiny white-ish line. That is a piece of a DNA fragment. When there is a line in there, that means there's a part of a deformable virus presented in that tissue. So you can see the brighter, the thicker the line is, means the more virus it is in there. So the gut has a lot of deformable virus. This is the control sample. But on the body, you can see there's deformable virus. On the spermatheca, there's also deformable virus. So they also tested the ratio of deformable virus versus the acting. That's a reference gene marker. You can see the difference. Basically, this is a quantified bar showing the intensity of the gel. And they also tested in the black queen cell virus as well as the SBV here. So the chronic bee paralysis virus, the CBPV, we see a little bit in the hemolymph and a little bit in the body. And the KBV here is showing only in the body part. So the vertical transmission of the viruses from the virus-infected queens to their offspring, the data is showing in this table here. And we can see on the top is definitely black queen cell virus and deformable virus. So this is come back to the beginning of the graph I have showed you. The vertical transmission is the route that viruses can be passed around in different developmental stages in the brood. And then within the worker bees, these vector-borne transmission can also be transmitted and passed around by the varroa mites. That's vector-borne transmission. By the food-borne transmission, that's through trophallaxis, the food feeding between each other of the worker bees, and the fecal-oral transmission, that's between the worker bees' fecal-oral behavior. So all these within all the workers, and this is called a horizontal transmission. That means within the colony for the same generation or the same group of bees. So next, I'll talk more about horizontal transmission. The foodborne transmission, that means if the honeybees eat the virus-contaminated food, for example, honey, pollen, royal jelly, all these are the food within the hive. So when they eat the virus-contaminated food, then they get the viruses. The oral fecal route is in densely crowded population, especially for black queen cell virus and deformable virus. So in a crowded population, they will do a lot of oral fecal behavior exchange. The contact, so when the worker bees all touch each other, you know, if they form a cluster, they touch each other, their wings, their hair, their body touching each other, that's a body physical contact. And they will transmit viruses from one to one as well. The airborne bacterial spores and viruses, so that's also another route. Like again, if I use the COVID example here, we know the COVID is one of the airborne viruses. The vector-borne transmission it's where the varroa destructor host. So almost, you know, all the American beekeepers know about varroa mites. That is the #1 enemy of everybody beehive. So varroa mites are and they can damage the colonies in the very very bad way, not just because they live in the capped cells and feed on the pupa, but also because they can carry the viruses around. So let's look at the bee development life cycle as well as the varroa mites. So in this figure, it's kind of busy slide, but let's go through one part by another. First, let's just focus on the bee. You see for 3 days are the eggs and then they hatch. So the green number days are the larval stage. The red numbers are the pupation days. So day 9 to all the way to almost 20. And this is the life cycle of honeybees. And of course, everybody knows this. But then let's look at the voramite because that is the vector to carry the viruses around. So the voramite get in at day 8 of the prepupa. And this female matured voramite can stay in that cell after the worker bees cap the cell, the voramite can reproduce. The first group, she will lay male eggs, reproduce male mites, and then the second batch, she will reproduce more female mites. And within the cell, the male female mites can again, the son and daughters of this mite can mate as well. So then they reproduce more. So by the day 20, the newly emerged worker, this is a day-old worker bee, when she comes out of the cell, all these mice, young and old, they had a party and they're all just gonna come out. And they come out and into the hive and they find another cell, they will jump in and hide again. So the cycle will go again. So when you look at this, the brood time is a green color for the bees. For the voramites, it's from the pupa time to the new adult time. So that is almost the longest time in the brood the mice can stay. So what viruses can voramite carry around? And we can look at, so these are the viruses on the list. They're ABPV, CBPV, IAPV. So IAPV is really acute paralysis virus, the slow paralysis virus, black queen, cell virus, the KBV, cloudy wing virus, the SBV, and deforming virus, DWV. And all these can cause the bee parasitic mite syndrome. So what you see is the colony is weak. It's infested with a lot of mites. So that indicate potentially a lot of viruses in there as well. For the biological vector-borne transmission, this happens when a vector is persistently infected with a pathogen, and the pathogen multiplies in the body of the vector before it is passed to another host. A biological vector may even be an essential part of a pathogen's life cycle. So the mechanisms regulating virus vector host interactions and transmission process of the virus are still really are still in a lot of people's research because this is very important. A lot of when people study viruses, it's really hard to only look at the virus or only look at the virus on bees. You have to include the vector of the voramites because the voramites play a very important here as a biological vector. So what about other vectors and factors besides voramites? Do we need to worry about anything else? The answer is unfortunately yes. Besides the voramites, there's also other potential vectors. For example, the Tripolali labs. These are the smaller mites right now hosting in Apis-cerana in Asia. So they are the future potential pest. USDA is watching very closely. They will try everything to stop this mite to get into USA. So deforming virus, one study has shown from 2009 that deforming virus are vectored by these small mites as well, by the Tripolali mites. So they run the similar gel like Judy Chen's paper did. You can look at design primers and run PCR and look at what kind of samples, and if they have, look at the samples of the mites and see if they have existing deforming virus. And the results showing all these highlighted lines showing bright white line are indicating they have deforming viruses in these mites. So except those mites, the wax moth are also bad. So the wax moth can actually be a vector of viruses. I think this is one part of information that maybe many people don't know about. So one study from 2016 has looked into this. They look at the wax moth larvae with the IAPV and the BQCV. So when they run the gel, they see these lines, even though they're kind of faint, but there is a line in there that indicated all these samples from sample 1, from sample 4, 5, 6, 7, 8, they all have IAPV virus. The 1, 2, 3 are probably control here. The sample 10 is the black queen cell virus, and sample 9 is a control. It's a positive control here. So besides the Lally Labs mites, the wax moth, small hive beetles are also a potential vector. People are looking into small hive beetles and to see if they are the potential vectors. And also ants are, for example, the Argentina ant is a reservoir for deformable virus from a New Zealand study. What about cockroaches and foraging on common flowers? So all these are very good questions, and we need more studies to look into these. So just one part to look at foraging on common flowers. The one possible route for the viral transmission is that when the bees, for example, honeybees go into the flowers to forage and collect pollen and nectar, and then other bees will also come to that flower, and other honeybees or other native bees or wild bees, and they can touch the flowers, the first honeybee with the virus touched, and they can get those viruses by that way. So for beekeeping management, what can we do to reduce the viral infection? That's probably the most important information for a lot of you here. As Dr. Tom Seeley mentioned from Cornell University, the Darwinian beekeeping may be one of the ways to rescue us. We really want to separate all the bee colonies in the bee yard. So if you have an apiary, you have a bee yard, I highly recommend everyone to separate your hives. The further out these hives are, the less drifting and less, visiting and mix from all the colonies. And that is also a reason why feral colonies tend to have a lower titer of viruses, because feral colonies are very far away from each other. They're up in the tree and they're by themselves. So yeah, so almost imagine, I can give example analogy, almost imagine you have 100 colonies on one bee yard, they're all next to each other. It's like a city, you're in a city urban area versus 100 colonies on 100 feral trees, and they're all very far from each other. So that's like 100 houses live in the rural area of Ohio. It's out of nowhere, and each house is by itself. It's very far away from other houses. So they have less visits from other bees from other hives. So second way is house your bees in small hives with less honey. That will reduce the disease and pest problem. But I know a lot of people probably don't want to take this otherwise, because they want to build big colonies. And so it's your own, judgment, and you can make a decision. But the small hives, they may help to reduce disease because there are less bees in there. The position of the hives, the higher off the ground, the better. Especially for these small hive beetles, the small hive beetles can climb, into the colony, honeybee colonies. But if you if you have the colony, the bee colony really high up, the bees can reach that height. But for the small hive beetles or the other pests, and they may not reach that height. Another thing to know is that only honeybees can do this hovering flight. They can land into the hole on a farrow tree colony, but for small hive beetles, they cannot do hovering flight. They have to climb on there and they have to go straight. They can only go forward. So the last advice is minimize relocations of hives. You don't move, so minimize moving hives around. That it's the same as the COVID practice, is minimize the viruses to be spread out in different areas. Basically minimize the travel of those bees. So in a summary, the viral transmission routes are very complicated. They can from vertical, horizontal, every way you can think of. So these vertical and horizontal transmission are the major routes for viral infections. The biological vector transmission introduces the X layer of complexity and make it more complicated. The ecological interactions and pesticide exposures may severe pathogenic infections in the bee colonies. So that's the first part of the talk. I think that's about 26 minutes. So the second part here, I'm going to talk about a new paper our lab just published recently about the end of last year. So this is a study we collaborated together with David Tarpey at NC State, Olav Ruppel at UNC Greensboro, and Mimi Strand and everybody, and Humberto Bo Cristiani. Now he's at University of Florida. So we look at the IAPV viral infection in the honeybees. So the IAPV virus is showing on the left picture. This is a viral variants. You can look at the viral particles under the electronic microscopy. So these viruses are the positive single-strand RNA virus. So just like coronavirus is also RNA virus. These Israeli acute paralysis virus are related to a colony collapse disorder. Used to think it may be the main reason for CCD in the 2008 paper, but later people have tested more, and this virus may not be the only reason. And the IAPV is spread out in the honeybee colonies. They are in the family of a Viridae, so they're dicystole virus. Because a lot of research and resources have been poured into the IAPV model system, and now this is a good system just to study the honeybee pathogen-host interactions. So we picked the pupa stage to study because the pupation time is so many days long. It's almost 10 days long, 10 to 11 days. And that is the longest time for bees to develop during the as a brood, and that's the longest time for varroa mite to hide in there and reproduce. So we are interested in the molecular mechanism. So when we talk about molecular mechanisms, that is down to the DNA sequence. So the genetic information in the honeybees are all in the honeybee chromosomes. And all these chromosomes, you stretch them out, really, they're like ropes all together. They're all double helix, almost like the blue balls showing here. So these are the basic structure of a DNA molecule. And within the DNA molecule, as the genetic material in the honeybees, there are 4 different types of bases. They're called A or T or C or G. So the C means cytosine. That's one of the 4 bases here. On the cytosine molecule, if on the position 5, there is a modification of a methyl group added in there. So that is a CH3 methyl group. After this modification of this methyl group, we call these cytosine methylcytosine. So these are methylated C or methylated cytosine. And why do I talk about this? Because this modification is very important. Even though we didn't change the sequence of this DNA, we add within the cell, based on the cellular signals, these modifications are added in. And this is called epigenetics. So epigenetics, epi means beyond. So it's beyond the DNA sequence change. Without changing the DNA sequence, there's still some cellular activities can actually manipulate or regulate the gene activities. So gene, yeah, maybe I should talk about genes. DNA are the genetic material I mentioned. So genes are basically pieces of DNA. And these different genes are different pieces, different fragments of a DNA. And they have functions. They carry different genetic information. Then they will produce RNA, they will produce protein, and then they will form tissues. So genes are very important for us to understand the molecular mechanisms. So just to help you understand why epigenetics are important, we can imagine all the genes are in a bee. It's like a bind together as a book. So I have a book here. So imagine all these are the genes of the bees. And it's just like those English letters I showed you, but for you, it probably doesn't make sense, or it's very hard to understand. What does it mean? It's just a bunch of letters here, right? But if we give them space and we have some punchline and you understand better, epigenetic modifications can be considered as the punctuation marks in the genome. A lack of prior knowledge makes the challenge greater. So because these space make them different words, and these different words suddenly, they make sense. We put it together as a sentence. So it's the same for this book, all the whole genome, all the genes of the honeybees, we add these, comma, period, the space. So all these can, are the modifications I just talked about. Because these formatting, spacing, and punctuation allows us to see the words, the sentences, and interpret the information that is there in the book. And the epigenetic marks perform a similar role in the genome. So what is a genome? I'm sorry about all the new words pop out of the slides. The genome is basically means all the genes of for example, we say bee as example, all the genes in a honeybee at one time from all the cells. So when we look at all the genes from a bee at one time, and all this together, a bee can have up to, I think it's about 100,000 genes or 150,000 genes in there. So all these 150,000 genes together, what's their gene expression level? What are these genes? That is called the genome. So for epigenetic studies, especially the DNA methylation, we've already know some information in the honeybees. On the left is a picture on the top of a worker and then the queen at the bottom. So there's a really famous study in 2008 by a Malaska's lab in Australia. They have tested the different queens, and by knocking down molecular manipulation of the bee larva, they realized when they knock down certain genes, these bees will develop into more queen-like. So they have control bees, worker larvae develop to worker, and they have queen-like, a few of them from the molecular manipulation. But when they have the targeted knockdown of this one gene called the DNMT3, it's a DNA methyltransferase 3, it's an enzyme for DNA methylation. And it's very surprisingly, they see many queen-like out of these larvae and less workers. So what that means, the gene is being manipulated and less gene in these larvae, and they all develop into queen-like. And they have a bigger ovary indicating the pictures. So what that means is reveal the function of demethylation. The knockdown of this enzyme decreased the level of demethylation and made those larvae into queen-like. So for our study, we're interested in the immune response and the gene expressions after IAPV infection. How do the DNA methylation play a role in gene regulations after IAPV infection? So this is how we design the experiments. We collect the wide-eye pupa from the hives. We have a PBS saline. This is a sham control. We inject the saline into the bee body. We didn't expect to see anything bad happen because there's no viruses in the saline. But we also injected IAPV viruses in those bees in the pupa, and we know this is a lethal dose of injection. So within 5 hours, the bees start to replicate in the bee's body, but the control ones should not have anything happen. The 20-hour, we know the IAPV virus would have a very significant impact on the honeybee cells. By the 48 hours, we know this is a lethal infection. So all these wide-eye pupa develop into red-eye or close to purple-eye pupa, and they are close to die, but they haven't died yet. So we collect these pupa and we extract the DNA out of them and we sequence. We can find out the information of all the DNA methylation marks, as well as their gene expressions and all the gene information. So now we're looking into the results of the experiment. The first three are the beginning 5-hour control. So these are the PBS saline control. We did see a high deformable virus in these pupa, but not much IAPV. So the second three group are the 5-hour infected honeybee pupa. We see some, almost 1/3 of the green color here, and these are the IAPV viral genes. And then we look at the 20-hour. So by the 20-hour, the IAPV infected pupa, we see very high expressions of all the IAPV viral genes. So more green means more IAPV viruses. And then by the 48-hour, they go up to almost 90 to 95%. So this is how do I say, this is a volcano plot. What that means is each dot here is one gene from a honeybee sample. So as I mentioned, total in the honeybee genome, we can see up to almost 150,000 genes. So there could be up to 150,000 dots on each little square. But when we compare the the control bees, and these are the P, so P5H means the PBS control after 5-hour injection. The I5H means the IAPV injected the pupa after 5 hours. So the more hours we go, the more red dots we see. That means the red dots means the genes that are significantly differentially expressed when we compare to the control. So we expect to see more genes to change dramatically. And so we expect to see more and more red dots on the later hours. I hope that makes sense. But I can explain more if you have questions. So we also compare the different hours. The one good approach for this experiment, for the experiment design, is a temporal comparison. We wanted to look at the dynamics of gene changes. So we have 5-hour, 20-hour, and 48-hour. The UpDEG means upregulated genes with their differentially expressed genes. So yeah, so DEG is the short name for a short name for the differentially expressed genes. And the differentially methylated genes regions are called a DMR. So we look at all the upregulated genes, only about 121 at the beginning, but then we see thousands, 2,000 genes differentially expressed after 20 hours, and then we see more after 48 hours. So all these are expected as we design experiment. However, we didn't expect the methylation patterns. We look at the methylation. They are either hypermethylated. Hyper means more methylation than control. The hypomethylation means less methylation than the control. So both hypermethylation and hypomethylation, we didn't expect to see more methylation changes at the beginning. And it's really bizarre to see later hours after 20 hours and the 48 hours, we see very, very few genes differentially methylated. So what are those genes? What do they do? And to answer that question, we did this gene ontology analysis. We look at all the genes. How are these genes similar to other genes in mammals or in Drosophila fruit flies? What are the biological processes they are in? So a lot of them are in the non-coding RNA processing, on the transfer RNA processing. So you probably wondered, what is RNA? So let me explain. So we talk about DNA. I hope you understand what's DNA. DNA is part of the genetic material, right? So all the genes begin from a DNA sequence, but the DNA sequence in the cell can be transcribed into RNA, and the RNAs are also fragments. But these RNAs can carry all these genetic information and tell the cell to make different proteins. So yeah, so RNA is another small molecule in the cell as well. So we also compared it to Jay Evans' research back in 2006. About 100, about a few thousand immune genes from G. Evans' paper. And we also compared to Christina Grozinger's lab's paper from 2015. Because the Gabriel et al. Did a study to look at the gene expression of the IAPV infected adult honeybees. So we did it in the pupa, so we want to compare. And we did see overlapping of a lot of immune genes from these two studies. So what about immune system? Because we're talking about viral infection, we're talking about immune response. two of the very important immune pathways are affected here. So one is the MAPK signaling pathway. Another one is the JAK-STAT signaling pathway. I don't want to go into too much details of these immune response. But if you wonder, honeybee immune systems are different from humans. Human immune system have two parts. We have the innate response, immune response. Then we have the adapted. So right now, everybody's getting vaccine for the COVID. That's the beauty of the adapted immune system, because the vaccine can stimulate the human body and produce T cells and remember what coronavirus is like. And then when you have real coronavirus, then they can produce quickly of an immune response to fight. But in the bees, they don't have adapted immune system. So in all the honeybees, they only have they only have the original primary, more of the lower kind of level of immune system. So so for them, only there's no T cell, there's no B cell, there's no vaccine for the bees. And what they do is they just use their signaling pathways, for example, MAPK or JAK-STAT or other pathways to fight the viruses. So we also look at the enzymes which are involved in the DNA methylation process. And those are two very important enzymes, the DNA methyltransferase 1 and the DNA methyltransferase 3. So DNMT1 and 3. We have looked at their gene expressions and we see a decreased trend. Both at 20-hour and 48-hour, we see less enzymes expressed in the bee's pupa. And that makes sense because less enzymes means less DNA methylation. And that could explain why the methylation levels are getting lower and lower. So very interesting. And this is the last part of the result. We really see a very interesting results of immune genes. There are 12 of these immune genes. We see their methylation changes first at the beginning of viral infection. And then we see their gene expressions and splicing patterns change after the 48-hour viral infection. So splicing is another type of gene activity. I won't have time to talk about it here, but if you're interested, I can explain more later. So all these genes are in different signaling pathways. For example, the hippo signaling pathway, FOXO, or the TGF-beta signaling pathway, or the mTOR. So all these are the innate immune response, just like in the bees. And they're part of the immune response. And it's very interesting. And this is a very exciting data or discovery we found in the paper from the research. They have methylation changes very dramatically at the beginning of the 5-hour, and then they have gene expression changes significantly after the 48-hour. So what does it mean? Why is this important? It means the methylation, the DNA methylation can actually change the gene expression in a delayed manner. So time really matters. For future studies, that this is a good first example to show bee researchers, excuse me, that it is very important to do time difference, different time points for research to look at viral infections at different time, and also look at the DNA methylation changes as in order to pick into a better picture, a more complete picture of the molecular mechanisms behind it. So one hypothesis is that the methylation can regulate the gene and regulate the immune genes and change their methylation marks, and this can give a signal, either block or induce the cells into cell death or apoptosis. And this has been shown in the cancer studies. So many of the genome-wide methylome and gene expression analysis link the DNA tumor virus infection to the dysregulation of host gene hypermethylation during the viral resistance and carcinogenesis. So the take-home message, do we see changes in the immune genes? Yes. What are the signaling pathways? They are JAK-STAT or MAPK signaling pathways involved. Do we see DNA methylation changes? Yes. We see a very unique pattern of the DNA methylation change as an immune response. And time really matters. So 12 of these immune genes act on changes on the DNA methylation first. And then it comes to their gene expression level changes after 20 and 48 hours. Right now, our lab has just started a new grant. This is a very exciting time. We just got a new USDA capacity building grant. And this is a collaboration with Reed Johnson at Ohio State University to study the pollen and pesticide residues in the honeybees, as well as Sandra Rehan. She's a professor at University of York in Canada. We're looking at different landscapes, different farm settings, conventional farms, organic farms, as well as the roadsides. How are these different farm settings, different landscapes affect the viral infection in the honeybees? How do they affect the pesticide residues and the health of the honeybees, as well as Serratina bees? So Serratina are the small carpenter bees here. So with that, I really thank you for your attention. I know there's a lot of information in my talk, and I'll be happy to help answer a lot of questions. And this research has been supported. The second part of IAPV is supported by a lot of funding and organizations. So this is our lab pictures. We have a lot of African American undergraduate students in the lab and doing really awesome research. And I encourage you to go come visit our Facebook pages and keep up with our news and research progress. So with that, I'd like to answer questions. Thank you.
All right, Hongmei. CK has a question. Do methylated nuclei acids code for amino acids?
Can you say it again?
Do methylated nuclei acids code for amino acids?
Not directly. They can affect the information on the RNA and the cellular process, the genetic activities from the DNA to RNA is one layer. And then from the RNA to the protein is another layer. So from the RNA information passed down to protein, that's the part involved amino acids. And that's the part is called a translation. So the DNA methylation is more worked on the beginning part from the DNA to the RNA. And that part of process is called a transcription. Yep.
Okay. Does anybody have any other questions? And while we're waiting for a few questions, would you like to go over the information that you have for your classes for Central State for the webinar series?
Yes. We starting next week, next Wednesday at 6 p.m. Eastern Time, Ohio time. We will start to have a webinar series. These are the webinars we designed for intermediate beekeepers who are interested in learning about queen rearing in Ohio or queen rearing in general. We are promoting sustainable beekeeping, and we would like to teach more people about queen rearing, about 48-hour queen cells, about how to increase their genetic diversity of your bee yard. So we have 6 talks. The next Wednesday, we first one, starting with Barbara Blaster. She's our Ohio State apiarist. She's our inspector of the state. She knows a lot about the certification process for queen rearing. If you want, you wanted to be a queen provider to sell queens in Ohio, and you want to do queen rearing, you want to hear her talk and talk about how Ohio regulate these queen rearing certifications. So you have to be certified to be a queen provider, and so that will help a lot of people to get information. And after next Wednesday, every month we have webinar talks on second Wednesday and the fourth Wednesday. So you can look at our calendar. It's in April, May, and June. Second Wednesday and the fourth Wednesdays. All of them are 6 p.m. The second talk will be from Mark Booker. He's a master beekeeper. He will talk about swarm traps, and he will talk about how to talk about 48-hour queen cells. The third and fourth talks are me talking about our research on the mite biting bees and our breeding program. And then Dorothy Morgan, she's also a very experienced queen rearing practice beekeeper, master beekeeper from Kentucky. She will talk about queen rearing in general, very good practice. And then the last 2 talks are from Ohio State University. We have Dr. Ree Johnson to talk about their drone research for drone reproduction. This is one of their new study. And then Chia Hua Lin, she's also from Ohio State University, Ree Johnson's lab, to talk about her queen research. So a lot of good talks. They're all very fascinating, very good speakers, quality speakers for beekeepers to listen. So I really encourage you to register. In order to register, you are welcome to go to our website. It's on Facebook. You just go facebook.com and /Central State University Bees. And you search, you can find our Facebook page. I can also type in here. And after you find our website, we have it pinned on all the way top. There's a eventbrite.com link. That's where you go to register. And from the eventbrite.com link, you will get the meeting link. Yep.
And we'll go ahead and post this on the OSBA website also for Facebook and website.
Thank you so much.
Not a problem.
all these webinars are supported from a USDA SARA grant. So we have the grant, we get supported from the grant. We really want to benefit Ohio beekeepers. So we design all these talks for you, for all of you. And I hope you really take the advantages of these opportunities.
All right, Steve's got a question. You said that the virus is getting into the honey. How safe is it for people to eat?
So yeah, there's probably just trace them out, and because these are bee virus, and they will not do anything on human, because the honeybees are the host for them and human is not. So I won't worry about the bee virus in the honey. Yep.
Okay. Nelda has got a question. She says, great talk with interesting findings. As this work continues, where might it lead? Viral treatment for honeybees or others?
Yeah, very good question. So as I mentioned, we have the new project just starting. We're going to look at viral prevalence of different viruses in the honeybee colonies in different landscape. So we're gonna look at different farms, like conventional versus organic versus roadside. How are the different viral prevalence in those population? And that will help us to understand better on the viral problem. And we also plan to design a new strategies and new approach to control viruses in the honeybees as well. But that part of work is still right now we're still in the planning. So we haven't, have any really good results or anything. So I'm not be able to talk about it, but definitely in the future, yeah, when we have, we're targeting the viruses, either deforming virus or the IAPV virus to look at how we can use molecular tools to do a better diagnosis.
Okay, Jason's got a question about your webinars. Will they be recorded and available later?
Yeah, that's a good question. So I think the webinar talks, not all of them will be published online. Only the speakers are agreed to for us to record and agree to, to put them on the website. We will do it. The first one, Barbara's, is not, because she said her talk cannot be online. So, once they be there, that will be our only chance to listen to her talk, and we will now post it online. But my talks and other talks, my talks will be online later, so it will be recorded. But Barbara's talk is not. All the other talks, I'm waiting for the answers from the speakers.
All right. And Anthony probably has the biggest question that it was on my mind also. How many of these viral infections are transmitted at the flower source or open feeding sites and apiary yards?
Yeah, that's a very good question. I think a lot of people are studying it right now. At least I know there are several papers published. They have looked at the honeybee viruses, how they are spilled over into wild bees, for example, bumblebees, other bee species. So there is a concern that a lot of viruses can spill over and got transmitted into other bees. And the potential very possible routes are the flower foraging, in foraging area and shared, foraging resources. For the second part, I think the person is asking about what open feeding.
Yes, ma'am.
Yeah, so open feeding and those bees, different worker honeybees from different colonies, if they have physical contact and physical touch, and that's again a way of a viral transmission as well. And I want to point it out, every year we have millions and millions of bees, honeybees, all managed honeybees going to Central Valley, California. For what? For almond pollination, right? I think almost all the people know this. And guess what? Think about all the school children. Millions of school children go to a same playground and they play. And same for those bees. All those millions of bees go into the California almond yards, and they will definitely have exchange of different viruses. Yep.
And that's all the questions we have for tonight. So thank you
Right.
very much for your time. And that was a lot of great information.
Yeah, thank you so much. I really appreciate the opportunity to talk all the Ohio beekeepers. Right.
Not a problem.
So yeah, feel free to send me email. Definitely if you have more questions, and I'm here all the time. So I have put the Facebook page of our university, our Bee Lab in there. It's a facebook.com Central State University Bees. And you can find the Eventbrite registration link. Yep.
Okay.
Hope to see you Wednesday.
Not a problem. Thank you, Hongmei.
Thank you. Have a good night.